I'm going to talk about killer whales, and it follows on a little bit from something that Dolph said in one of his responses, looking at niche variation. And Dolph mentioned work done by Dan Bolnick, where he's been looking at niche variation in stickleback populations. And niche variation is where, in a generalist population, so rather than having two specialized populations, you have a generalist population, but instead of all individuals consuming all resources equally, the niche width of the population is made up by individual specialists. And these individual specialists can be subject to disruptive selection. So, for example, things like the beak size due to the seeds that they feed on in the Darwin's finches. And if these traits that are linked to the ecology are also linked to some kind of reproductive isolation mechanism, then it's hypothesized that this niche variation could be an early stage of sympatric speciation. So two sort of thoughts with that, so two provisos that would need to be there for that to occur would be for this niche variation to be present over evolutionary timescales, and also for there to be a trade-off, some sort of constraint that reduces the fitness of an intermediate phenotype. So the model I'm working on is killer whales. So I always feel a bit embarrassed at these speciation conferences because they're not very model organisms and kind of difficult to work with. But they do form these distinct ecotypes in certain parts of the world. And the most famous are in the North Pacific, around British Columbia, for example, where you have a mammal-eating form and a fish-eating form. And studies that myself and others have done suggest that there's reproductive isolation between these two forms, the mammal-eating and the fish-eating form in the North Pacific. So, for example, using complete mitochondrial genomes, there's complete lineage sorting, which suggests that there's either no dispersal or it's very, very limited and we're not detecting it. And also using nuclear markers, so microsatellites have been used in the past, and Phil Moran's just generated a data set with 100 SNPs from across the genome. And again, that's suggesting very low admixture at nuclear markers as well. And we've actually just, we're just doing whole genome resequencing. We just got the sequencing data back as well, so we had to look at that further. So it's suggesting that these two ecotypes in the North Pacific are at a very late stage of speciation, perhaps even completion. There's a steep step in the ecology, the strong lineage sorting of the mitochondrial genomes, and also strong genotypic clustering, suggesting that there's some kind of reproductive isolation mechanism there. And I should also say that this backs up what's been observed in 40 years of field studies on these animals, where there's been no interaction seen between the two different ecotypes. In fact, they actually often get seen to actively avoid each other. There is also evidence for a constraint on an intermediate phenotype, and that's the hearing levels of their preferred prey. So, for example, if you're feeding on a marine mammal and you're a killer whale, your vocalizations are going to be detected by that prey. So basically, this is a harbour porpoise and a harbour seal hearing curve or an audiogram, and they can detect everything above this threshold. And this is the decibel levels across this frequency range of a killer whale recorded at approximately half a kilometre away. So they would already be able to easily detect killer whales at that distance. Whereas salmon have very poor hearing and it's very low frequency range, and they wouldn't actually be able to detect killer whales until they were very, very close by. And so that would suggest that possibly being an opportunistic hunter is quite difficult for killer whales, especially as well as fish-eating killer whales are sort of hypothesized to actually use the calls to coordinate the group during hunting for fish. And that's reflected in the call rates when they're foraging. So, for example, the fish-eating killer whales produce calls quite often when they're hunting fish, especially salmon or herring in the North Atlantic. Whereas the mammal eaters, they tend to hunt in very small groups, and they're almost completely silent when they're hunting, and they actually just vocalize once they've caught something. Now, we did some work a couple of years ago using the mitochondrial genomes, and we did various different phylogeographic modeling. We used IMA and a number of other tests, and all of them I came to the same kind of conclusion, that these fish-eating killer whales in the North Pacific actually came from the North Atlantic back into the Pacific. So they split from the mammal eaters around 700,000 years ago and then came back into secondary contact around 120,000 years ago. And so maybe that was what generated these two different specialists, possibly. Maybe they'd already started to specialize in allopatry, or maybe it was some kind of reinforcement on secondary contact. But it does beg the question, could this divergence occur in sympatry? And we have quite a good model for that in the Northeast Atlantic, or a good study system to test it. We also have, this is actually two animals that have stranded around the UK in the last few years. We have two different ecotypes in these waters as well. And again, it seems to be from secondary contact with sort of highly specialized, larger form that's come up from the Antarctic, and then the more common Northeast Atlantic form, which has really worn down teeth in almost all adult cases. But this is what I'm going to talk about today. We found quite high variation in the stable isotopes of this form with the worn down teeth. And we wondered if that had the potential to basically be the starting point of sympatric speciation. And just to sort of show that in a bit more detail here, we have two examples. They're both from the coast of Denmark in the last 150 years. They both have worn down teeth, and they have identical mitochondrial DNA control region haplotype, but really different stable isotope ratios. So the N15 kind of gives you a rough idea of the trophic level they're feeding on, with this one here having kind of more fish-based diet, and this one here having a more mammal-based diet. And there are also fish bones in the stomach of this one, and this one actually had the remains of 13 harbour seals and 13 harbour porpoise in its stomach. And actually it choked to death. It actually got a seal lodged in its windpipe, so had eyes too big for its stomach. So while killer whales aren't exactly a model organism to work with, they do leave quite a useful trace of their past historical biogeography behind them, in that they're a big animal and they leave a nice subfossil record. And we have quite a rich subfossil record in the North Atlantic, and in particular the North Sea. So we've been collecting those, and they basically range over the last 10,000 years, since since the start of the Holocene, when the glaciers retreated. And so here, for example, we have some subfossil teeth. They're also worn, just out like these two, and they're 7,000 years old. And we can get DNA from those, and also stable isotopes. So the aim of this study was to use ancient DNA to basically confirm the species and make sure it belonged to the same lineages as these modern killer whales we were looking at, to use stable isotopes to estimate niche width, and then basically to look at the genetics of the modern specimens and just establish the evolutionary outcomes of niche variation. And the samples have come from a relatively small area, so we can basically, this is the kind of distance that killer whales can travel very quickly in a couple of days, and their home range would easily constitute that area. And so we're sort of hopefully ruling out any kind of geographic variation in the resources that are available. So this is the stable isotope values of the more recent samples, and it's based on tooth and bone, which are basically tissue that are accumulating over a long time span. So we're getting a dietary signature of multiple years with these stable isotope values. And these samples, I should say, they're still quite old. They're up to 200 years old, so up to just 10 years old, but they're not really recent ones. But you can see they span from the low trophic level prey of herring up to the high trophic level prey of a harbour seal. And some cluster over here as well. And just as for a comparison, we have the stable isotopes of skin samples from Norwegian herring-eating killer whales during a period. So skin will give a much shorter time signature, and it's a period when the herring are really amassed in the Norwegian fjords, and the killer whales are pretty much just eating herring, just to give you an idea where that is. But they're feeding on Norwegian herring, which is a little bit more offshore than North Sea herring, so hence the slight difference in the carbon signature. When we put the subfossils in, we get almost the same pattern. They basically span that broad niche from low trophic level to high trophic level, with basically no sort of obvious bimodal clustering. It seems to be fairly continuous between the two prey items. And we've been using this cool new method, this Bayesian ellipse method, which is part of the SIAR package for stable isotopes. It's very neat. And there is a significant expansion in the niche width when we compare the subfossils to the more recent samples. And that seems to be pushed by this this increase in the carbon, which we actually seem to think comes from more Norwegian animals moving into this North Sea area. But across both time periods, there's quite a broad niche width. So what we're wondering is that there the raw material basically for sympatric speciation? Well, when we looked at the more recent samples, we used target enrichment capture methods to produce whole mitochondrial genomes. Because, of course, these samples were still 200 years old in some cases. And we got some evidence of lineage sorting. We mapped the stable isotopes over the mitochondrial DNA. And this is just the nitrogen values shown here. And you can see that within a clade, most individuals, in this case, are feeding at a fairly low trophic level. But in other clades, there's evidence for individuals that have changed quite dramatically in their diet. So, for example, here, there's one individual in this clade feeding at a higher trophic level than the rest of the clade. And in the grey clades are ones where we didn't get stable isotope values for them. But basically, it suggests kind of weak to intermediate lineage sorting. And then we use microsatellites on modern samples. We'd previously done a population genetics study in the northeast Atlantic, and we found that population structure was mostly determined by which fish stock they were hunting. So, for example, herring and mackerel and then tuna. And because the fish basically don't overlap spatially very much, it led to population structuring. And we looked at animals where they'd stranded or they'd been caught by hunters, and they had seals in their stomach. And it was quite a small sample size. Unfortunately, it's quite difficult to get this data, but they showed complete panmixia with the herring eaters. So there was really no obvious indication of any kind of reproductive isolation based on a mammal versus fish-eating dichotomy. So I'll have to go quite quick. Oops. And but it really suggests that they're at the very kind of inception of this speciation process. So really got a shallow ecological decline, weak lineage sorting, and really reproductive isolation seems to be, absent. Just very quickly, in the Northeast Atlantic, we've done field work with these animals, and they show parallel phenotype to what we see in the Pacific when we're looking at the ecotypes. So the fish eaters call, the fish eaters produce calls frequently when they're hunting fish, and the mammal eaters stay mostly silent. And also the same pattern with the group size. So fish eaters form an array of different group sizes, and mammal eaters usually travel in small groups. But unlike the Pacific, we see animals switching between these strategies. And so while opportunistic hunting might be a low fitness intermediate phenotype, perhaps being a generalist that switches between these specialist strategies, there's less of a sort of a penalty for that. So, for example, this animal here was seen feeding on herring in a large group off of Iceland in the 90s, and repeatedly so. And then this animal here has been repeatedly seen. In fact, this animal actually got photographed last night off of the north of Scotland killing a seal. And again, it's always in a group of 6 when it's doing that. And so, in terms of why I think there might be this difference, is that these changes, these things like group size and vocal rates, they're plastic responses. They're not really adaptive genetic divergence. And so it probably means that if the divergent selection is mainly acting on these plastic responses, it doesn't really limit gene flow between them. And so it's quite a sort of weak link between the ecological selection and any kind of reproductive isolation. And so just quickly in conclusions, I've written far too much there, but just to quickly summarize it, I guess what this shows is that killer whales span this sort of speciation continuum. And I think that's something that's coming up more and more as people look at more and more study sites, that they find varieties from very discrete phenotypes to continuous phenotypes and kind of everything in between. And also, I think this study shows that it's kind of difficult for this sympatric speciation due to, say, niche variation to basically move forward into completion. And thanks very much. And that's just my co-authors.
Thank you, Andy.
No problem.